Oncovin. Tubulin is a protein that makes possible such vital life processes as cell division and the movement of materials within cells. In contrast to intermediate filaments, which are composed of a variety of different fibrous proteins, microtubules are composed of a single type of globular protein called tubulin. Tubulin is a dimer consisting of two closely related 55 kD polypeptides, alpha-tubulin and beta-tubulin. Like actin, both A and B tubulin are encoded by small families of related genes. In addition, a third type of tubulin, Y-tubulin, is specifically localized to the centromere, where it plays a critical role in initiating microtubule assembly. Tubulin dimers polymerize to form microtubules, which can generally consist of 13 linear protofilaments assembled around a hollow core. The protofilaments, which are composed of head-to-tail arrays of tubulin dimers, are arranged in parallel. Consequently, microtubules, like actin filaments, are polar structures with two distinct ends, a fast-growing plus end and a slow-growing minus end. This polarity is an important consideration in determining the direction of movement along microtubules, just as the polarity of actin filaments defines the direction of myosin movement. In microtubules, GTP hydrolysis also results in the behavior known as dynamic instability, in which individual microtubules alternate between cycles of growth and shrinkage. Whether a microtubule grows or shrinks is determined by the rate of tubulin addition relative to the rate of GTP hydrolysis. As long as new GTP-bound tubulin molecules are added more rapidly than GTP is hydrolyzed, the microtubule retains a GTP cap at its plus end, and a microtubule growth continues. However, the rate of the polymerization slows. The GPT-bound tubulin at the plus end of the microtubule will be hydrolyzed to GDP. If this occurs, the GDP-bound tubulin will dissociate, resulting in rapid depolymerization and shrinkage of the microtubule. Tubulin is a heterodimer protein, meaning it's comprised of a pair of polypeptide chains called monomers that differ in the sequence of their amino acids. Each tubulin monomer, the alpha and the beta, is a compact molecular structure with three functional components or domains: one that binds to nucleotides, one that binds to drugs like Taxol, and one that looks to be a binding site. Microtubules have the ability to shift through various formations, which is what enables a cell to undergo mitosis or to regulate intracellular transport. The formation shifting of microtubules is made possible by the flexibility of tubulin, which is why scientists have sought to understand the protein's atomic structure since its discovery in the 1950s. Researchers first polymerized tubulin proteins under the same conditions in which microtubules are formed, except for the additional presence of zinc. The zinc prevents tubulin chains from curling around into hollow fibers. Instead, the polymerized tubulin forms two-dimensional crystalline sheets that are ideal for imaging by electron crystallography. The use of electron-based rather than X-ray-based crystallography techniques customarily employed in protein studies was crucial. Interest in tubulin structure heated up intensely in the recent years when Taxol, a natural substance found in the bark of the Pacific yew tree, was known in clinical tests to be an effective treatment for a number of cancers, including ovarian, breast, and lung. Cancer occurs when cell division runs amok. By binding to tubulin, causing the protein to lose its flexibility, Taxol prevents a cell from dividing. With better knowledge of tubulin structure and its interaction with Taxol, scientists believe that an even more effective anti-cancer drug, one that interacts only with tubulin of cancerous cells, could be synthesized. Cholecystine and vinca alkaloids are two of the first microtubule destabilizing agents to be discovered. These two compounds depolymerize microtubules by interacting with various B-tubulin sites. In particular, vinca alkaloids interact with tubulin at specific binding sites which differ from those of other agents, including cholecine and taxanes, interfering with microtubule dynamics, blocking polymerization at the end of the meiotic spindle, and leads to metaphase arrest. Thanks to their particular mechanism of action, vinca alkaloids have been widely used in anti-cancer therapy, usually in combination with other chemotherapy agents which do not have cross-resistance with them. First-generation vinca alkaloids, such as vinblastine, have been included in the treatment protocol of both Hodgkin's and non-Hodgkin's lymphoma and testicular carcinoma, while vincristine has been approved for several years in the treatment of hematological tumors such as acute leukemia and multiple myeloma, but also rare tumors such as rhabdomyosarcoma and neuroblastoma. However, vincristine treatment was associated with a severe neurotoxicity, while the suppression of the bone marrow was more frequently reported during vinblastine therapy. Second-generation semisynthetic vinca alkaloids, vinorelbine and vindicin, have shown a broader spectrum of anti-tumor activity in vitro, along with decreased neurotoxicity. Vinorelbine was approved as single agent and in combination therapy with the treatment of both hematological and solid tumors, including lung, breast, and gynecological tumors. Recently, another synthetic vincoalloid, vinflunine, has been approved in Europe for the second-line treatment of metastatic urethral carcinoma. It is the first fluorinated microtubule inhibitor, which was associated with higher anti-tumor activity than other vincoalkaloids, showing also an excellent safety profile. It prevents the formation of spindle fibers during metaphase of mitosis. Vincristine is an alkaloid associated from the Madagascar periwinkle Catharanthus rosus, formerly classified as Vinca rosea, which led to it being called a vinca alkaloid, and thus vincristine. It binds to tubulin, the protein that makes up the spindle fiber microtubules, and prevents metaphase of mitosis.